there and catch up with your PhysioX, or come there. I'm converting those hours, those 3 hours, to office hours, so I will be there. I'll be sitting in lab, so if you want to come and do your PhysioX there, you're more than welcome to. But since it's only PhysioX scheduled for next week, you don't really need to come in. Okay, so next week is kind of a little break for you, so use that to catch up. Okay, please catch up. The lab exam was a doozy for people today, so but who knows, maybe Thursday's class will do well. But I have decided to drop one lab exam and one lecture exam, okay, towards the final grade. So your lowest lab exam and your lowest lecture exam will get dropped, okay, which puts the extra credit exam in jeopardy, okay, because then that just becomes grade inflation. I can't really do that. So I have to kind of think through that, get the dean's blessing on it, and then if she says, okay, since it's only 25 points, I don't think she'll have much trouble with it. It's only a quarter of an exam, so I don't think she should have trouble with that, but I'll keep you posted, okay? But I'm definitely dropping the lowest lab exam as well, okay? And the justification for that is there's a lot of labs to be testing on all at once, okay? Next semester I have to redesign the syllabus to include more frequent lab exams. The problem is I don't have the time, right? I have to cut into lecture to put more exams, right? And I can't afford that, so I'm gonna have to do, but that's my headache, I gotta deal with that. Any hoops? Everybody has the kidney notes ready? Yeah, so next week is optional. We will have lecture, so lecture is gonna be as usual, but the lab session next week is optional, so you don't have to come if you don't want to. Or you can spend that time at home and do your PhysioX experiments at home. You don't have to be here. I'm trying to save you a trip. Although you still would like to come to the lecture, then you have to come to the lecture, okay? All right, so we're dealing with the urinary system. The urinary system is very complicated. It is part of the excretory system. It's a major part of the excretory system. The other components being the lungs. The lungs are part of the excretory system. The skin is part of the excretory system also. And so is the liver. They're all part of the excretory system. And the urinary system is one part of it. So we're going to focus heavily on the urinary system. We're going to first look at how urine is made, and then we're going to go into the more important part, which is fluid balance. We're going to be looking into how our body maintains electrolytes and how our body maintains water balance. So it's really, and pH balance. Extremely important chapter. Okay. The kidneys are bean-shaped and retroperitoneal, meaning it's in the back. Kidneys are in the back. Kidneys are not in the front, they're in the back. Okay? And the renal hilum is where the blood vessels enter. Okay? So that's that little curvy shape of the kidney bean, okay, portion. And the adrenal glands rest above the kidney. Ad means on top of. So adrenal. Renal means kidney. Ad means on top of. So adrenal glands are above the kidney. Typically, you have three regions that are important, and the first two are the ones that are important for us. The cortex of the kidney. Okay, this is not the adrenal gland, so be careful. That's not what we're talking about. We're talking about kidney. Kidney has the same kind of regions as the adrenal gland does. You have the cortex, you have the medulla, and you have what's called the renal pelvis. So you have the renal cortex, the renal medulla. So when you're a medical professional, you should never ever say cortex medulla of what? Right? So you have to be very specific. Ovarian cortex, ovarian medulla. Adrenal cortex, adrenal medulla. Cerebral cortex, right? And then medulla oblongata. And here we've got renal cortex, renal medulla. So you have to be very specific. The renal cortex shows high vascularity. So one of the reasons this chapter is interesting / heavy, it's heavily interesting, is that the anatomy and the physiology are inseparable. Okay, you cannot, there's no way, I don't think I've seen a teacher separate the two beautifully, okay? Because when you had me for anatomy and I taught this chapter, it was impossible for me to not bring in some of the physiology. right? It carries no meaning. It becomes completely meaningless. And in physio, it's impossible to teach it without the anatomy. Because unless you know the structure, you have no idea where this is all happening. Seems all abstract, right? So you kind of have to know both. So it makes it really interesting, but it makes it doubly heavy. Okay, because you're studying two subjects at once. So you have high vascularity. The cortex appears really, really red for two reasons. The cells are very rich in mitochondria. This should not be surprising. You saw the same thing in muscle, correct? Muscle is really, really red because it has skeletal muscle. Really, really red because it has a lot of mitochondria, particularly slow oxidative fibers, correct? Yeah. Same analysis here. The renal cortex is really red because it has a lot, these cells have a lot of mitochondria, which should immediately tell you something very, very important. This is also why this area of study is very heavy, because every statement has 10 different things hidden behind it, and you have to go explore it. So why is the renal cortex red? Renal cortex is red because it receives a lot of blood. Well, why the heck does it receive so much blood? Because these cells need a lot of energy, which explains why it has a lot of mitochondria. Correct? Mm-hmm. Which means the energy expenditure of these cells is very high. These cells have a high turnover of ATP. Am I making sense? Mm-hmm. Just like skeletal muscle, they use a lot of ATP. And I can make life a lot simpler for you by simply stating these cells do a ton of active transport. Remember active transport? I told you, no cell transport, bye-bye kidneys. Okay, so you need to know the cell transport chapter. So go back and watch those videos, extremely important. The medulla is in sharp contrast, pretty white in color. The medulla does not have a lot of ATP use, and you will see what the medulla is used for. The medulla is there primarily to help absorb as much water as possible. Okay, remember we are land animals, correct? So for us, water is precious. We cannot lose water, right? We push 180 liters of blood through the kidneys every day. 180 liters of blood, that's a lot of blood going through the kidneys. We pee less than a liter. Okay, so what's happening to all that water is being absorbed back. Are we clear? And when you're dehydrated, it's even worse. So the medulla shows a lot of renal pyramids, okay? And pyramids are shaped like little triangles, right? Like that. Separated by renal columns. Now this anatomy you can ignore. I don't really care if you know this or not. But the pyramids are made up of nephrons. I've told you anything on means a unit. Osteon is a unit of bone. Okay? Neuron is a unit of nerves. Correct? A photon is the unit of light. So a nephron is the unit of the kidney. Because the word nephros in Greek means kidney. Nephros means kidney. So the bottom line, you can ignore all these calyces. So put an X through all this. You don't need all this. This is all anatomy that's not really relevant. So I'm going to head right into the last sentence. It's important. Urine that is made is moved by peristalsis. This should not be surprising. You guys know what peristalsis is, right? Rhythmic squeezing, correct? By the way, your next lecture exam is not next week, but the week after. And it's only going to cover respiratory and digestive. Okay? Only respiratory and digestive. Those two review sheets are already up. Thank you, Fran. She's nagging me to put that up. So those are up already. So please study. Okay? The review sheets for respiratory and digestive are up already. Yes? Mm-hmm. So please start studying for your next lecture exam. This is the time to improve your grade. Okay? So I'm purposely excluding kidneys out of the next exam. So where will kidneys be? It will be on the exam after that. Okay? Most likely, we will not have time to do reproductive. Most likely. Okay? We'll run out of time for reproductive. So pretty much the last exam is going to cover kidneys and immune. Okay? Both are fairly heavy chapters. Okay, so that's going to be on the final, okay, or that last exam, not the final, but the last exam. But you have plenty of time to study for it. Okay, so start studying now. All right, so vascularity, which is blood supply. Okay, this you can ignore. This is for anatomy. This you can ignore. However, one point I want you to understand is the kidneys have a very weird system. Okay, kidneys have a really weird system. Notice. So I don't need you to know arcuate arteries and segmental and all that. I don't need you to know that. Where I want you to focus is the cortical radiate. So starting from the cortical radiate, well, one thing I want you to know is this is a very intricate system of arteries, right? Makes sense. Kidneys need to be filtering blood, right? So a lot of blood goes there. More importantly, so anytime anyone asks you what is the function of the kidney, and you say it's to make urine, it's completely wrong. Okay, the function of the kidney is not to make urine. Are we clear? Mm-hmm. Making urine is a side effect. It's really a very peripheral side effect. The main purpose of the kidney is water balance. Water balance. That is the main function of the kidney. Okay, so please understand that, because if you don't get that internalized, everything is going to be all warped up. So understand that the main function of the kidney is water balance. So from the cortical radius, the afferent arteriole, so from an artery, you get arterioles. This should not be a big surprise, right? From arteries, you get arterioles. From the afferent arteriole, you go into the glomerulus. You need to know this to do the physio-X. And the physio-X is going to be really tough for the kidneys, okay? So please make sure that you do it very carefully. That's why I don't want you wasting time commuting here. Okay, I want you to spend that time on the physio-X. Okay? And basically then you have going into the glomerulus. Glomerulus is a capillary bed. Okay? Arteriole to capillary. No surprise, right? This is how it happens everywhere. Correct? You go from artery to arteriole to capillary. Capillary then goes into venule, venule into vein, right? And vein back to the heart. Right? Do you remember the circulatory pathway? Do you remember the circulatory pathway? Yes. Yes. So that's what's happening, except you have an extra loop. You have an extra loop. We saw this kind of an extra loop with the liver. That was the hepatic portal vein, right? So here you see an arteriolar loop. You're looping the arteriole, where you go from afferent arteriole to glomerular capillaries, back to the efferent arteriole, down to the peritubular capillaries, and then onto the venule. So you have two capillary beds serving this. Makes a lot of sense, because you need two capillary beds to do filtering and reabsorbing, as much reabsorbing that you can do. So this kind of a system is called a portal system. Portal system. It's an arterial portal system. Arterial portal system. And the peritubular capillaries, or vasa recta. Everybody knows what recta means, right? Straight, right? Those are vessels that come down straight. Vasa means blood vessel, recta means straight. So vasa recta. And the vasa recta then dump into the cortical radiate vein. Okay, so then it goes into the vein. Okay? And you can ignore the rest. So the ones you need to focus on in terms of these vessels are only the ones I'm going to discuss later. Okay, which is you're dealing with the afferent arteriole, glomerulus, efferent arteriole, peritubular capillaries. Make sense? So those are the only ones I need you to focus on, because other than that, we're not going into any other vessels or mechanism. So those are the important ones. Extremely important point. Weirdly enough, and I think we have this again tells us that evolution is still going on in our body. Only the sympathetic innervates the kidney. Okay? That does not make any sense. We really should have parasympathetic going there too. But as of now, only the sympathetic. Okay, give it another million years, maybe the parasympathetic will catch up. But as of now, only the sympathetic innervates the kidney. I will ask this. This is an extremely important point. Only the sympathetic innervates the kidney. And remember all those receptors, right? Alpha 1, alpha 2, alpha 3, beta 1, beta 2, all that stuff. You need to remember that. So please go back to that table. You need to look at which ones relate to the kidney. And you need to know them. I will ask the adrenergic receptor type. You will need to know adrenergic receptor type for your next exam. And epinephrine or norepinephrine usually vasoconstrict the arterioles with the word of caution. Okay? Afferent arterioles act like normal arterioles and they will vasoconstrict. Efferent arterioles exhibit exactly the opposite behavior. So sympathetic will vasodilate the efferent arterioles. Are we clear? So afferent and efferent act oppositely. This is really important. And this is the main point of PhysioX next week. Okay? Is the afferent arterial diameters. Kidneys have about 1 million nephrons. I don't really care whether you know this or not, anatomy. They have 1 million nephrons, thousands of collecting ducts, and so you're collecting urine from millions of these filtering structures. Okay? So before I move on to nephron structure, which is extremely important for nephron physiology, before I go into the nephron structure, look through these notes and see if there's anything I can clarify. I will get back to those notes in a minute, but I do want to start off with this diagram. First of all, a nephron is a multi-unit structure. Multi-unit meaning it has thousands and thousands of cells. It's not a single cell like the neuron. Neuron is a single cell, correct? Here you have thousands and thousands of cells. Okay, it's a big structure. A nephron is huge. Okay, and the nephron receives blood vessels. Okay, and it is a tubular structure. It's a tube, and so any tube you will have a lumen. Correct? Remember what a lumen means. The word lumen is really important here. Lumen means the hollow of a tube. So you will hear me say luminal. You will hear me say basolateral. Luminal and basolateral. Luminal is the side facing the hollow of a tube, and basolateral is facing the opposite side, which is facing blood. Okay? So if this is your tube, your kidney tube, right? This shouldn't be surprising. We didn't digest it. It's just like that, right? So the hollow, the inside of the tube is your lumen. Okay? So if I have a rectangular cell, my thumb is facing the lumen, my other fingers are facing the blood. Yes? So what are we doing? We're absorbing things transcellularly. Correct?
Yes.
So we're taking everything in and we're absorbing whatever we want. Except here, it's called reabsorption. Okay? Because you're not really digesting and absorbing, right? It's already there in blood. You're filtering everything from blood and then you're taking it back. So reabsorption. Is that making sense? Now, question is, why did we evolve something like this, right? Why can't you, look, if you don't want something, you throw something in the trash, right? Or a bunch of clothes that you don't want. You collect them all and then you take it to the Salvation Army or wherever, Goodwill, right? And you give it to them and you're done, right? You don't give your entire wardrobe to Goodwill and then take whichever clothes you want back. That's tacky. Nobody does that, right? The kidneys are tacky, and for a very good reason. So what we do is we dump everything we have in blood into the kidney, and then take up whatever you want. The reason is, and this is the best way of doing this, because honestly, if you did it the other way, you would save so much energy. Correct? This is really putting a lot of effort in there, right? It seems excessive. But the important reason why this happens, and a lot of organisms, including worms, do this. Okay? Insects have Malpighian tubules. Worms have very primitive nephronic helix, okay? But they all work this way, okay? So, question is, how come, right? The reason is, what is trash now? In a couple of seconds, is going to be treasure. So, trash versus treasure. The perspective changes every few minutes. I'll explain. So take me for example. I ate a fairly decent breakfast this morning, eggs and waffles and whatnot, right? So I ate quite a bit. And right after I eat, sodium was probably excessive in my blood, correct? And at that point, sodium is trash, needs to get away from my blood. But right now, after my intermittent fasting for 8 hours now, right, in my hangry state, what is probably happening? Sodium is probably really precious, right? So what is my kidney trying to do? Is trying to keep the sodium in my blood. Am I making sense? So what was trash at 8 a.m. is now treasure. It gives kidneys that flexibility. Okay? So when you filter everything down, okay, at that moment you can pick and choose. Are we clear? Mm-hmm. The reason is a lot of parameters in your blood change very, very quickly. And when that happens, kidneys need to dump everything down and then pick up whatever it needs at that point of time. There are some things which are purely trash. Urea is trash. Nobody wants urea back in the body. Urea is gone. Okay? Even though we do pick up some urea, but urea is trash, and that's what it is. Our urine has urea. That's why it's named urea. It's in our urine, right? And it's a fertilizer for plants. Okay? So urine has urea, so urea is an excretory product. Urea is a goner, right? So there are a few things. Steroid metabolites, we don't want them back in the body, right? So we get rid of it. So there are some things which are definitely excretory, you want to get rid of them. But everything else, the body believes in recycling. Are we clear? Mm-hmm. So if it is needed, you want to take it back. You want to have that ability or the choice to take it back up. Make sense? That's why evolution picked this kind of a kidney function. Make sense? Mm-hmm. So what is the working principle of the kidney? You filter everything from blood that is smaller than a protein. Proteins are never filtered into the urine. You never want that. So everything smaller than proteins is filtered in. And you've already seen this before, correct? Remember capillary exchange? I told you, everything comes down that's smaller than a protein, right? And then osmotic pressure pulls it back up. Same exact principle here. You do exactly the same thing here, okay? Except the taking things back is very, very complex, right? Because you need to be reabsorbing a lot of things, right? And not only that, the kidney employs both transcellular and paracellular pathways. Kidney employs both, unlike the digestive system. Remember, in the digestive system, there is no paracellular. Okay? Here, things move between the cells too. Make sense? Because the tight junctions are fairly leaky. They're not perfectly tight. And in fact, tight junctions were discovered in the dog kidney, okay, as a concept. So what is the structure of the nephron? This is extremely important. Anatomy of the nephron is crucial. So you have a little bowl, okay, that you start with. This thing is not very anatomically accurate, but it doesn't matter. I think it's a really good diagram. So we have this right here. And again, you don't really need to know the structures of all of these, that's for anatomy. But you have a bowl here, okay, and that's called Bowman's capsule. Okay, Bowman's capsule basically houses the capillary bed called the glomerulus. The glomerulus sits there. So when blood comes in, with fairly high pressure. Now I want you to, see here's again relevant anatomy. So all the renal artery branches right off of the descending aorta. And you know how much blood goes through the aorta, right? All of it, right? So renal artery gets first dibs, right, on that blood. So a lot of blood comes in with a lot of pressure. And if you damage those glomerular capillaries, it's going to be bye-bye time because nothing's coming back. Just like the lung, probably will come back, but very slow, right? So you lose, these things need to last you your whole life, okay? This is another reason we have two kidneys, right? Compensatory, right? So, too important. So blood goes in there and pushes itself against the walls. These are fenestrated capillaries. These are fenestrated capillaries, meaning they have holes in them. So when blood is pushed, everything comes out. So you have size exclusion. Size exclusion. That means anything you're filtering based on size. Okay? What is very, very interesting here is that the Bowman's capsule also has a high negative charge. Now, in general, proteins are negatively charged. In general, proteins are negatively charged. So if you have a highly negatively charged capsule, that will also repel the proteins and prevent proteins from getting into the kidney. So you've gone through millions of years of evolution to keep proteins from entering these tubules. Are we clear? Okay? So if you're going to be unhealthy and irresponsible and be diabetic and your sugar is not in control, eventually you have so much damage to these capillaries that proteins come through. So if you have protein in your urine, chances are you have urinary tract infection. Okay? Chances are it's urinary tract infection. But very unlikely if you have protein in the urine, and you did that lab last week, right? If you have protein in the urine, it could be diabetic nephropathy. Diabetic nephropathy. If you keep your sugars uncontrolled for well over 10 years, it takes 10 years to damage these things. Minimum 10 years. Okay? These are that strong. Okay? I mean, my sugar has been out of control for 15 years. My kidneys are working perfectly. Right? Absolutely no protein in my urine. Okay? So I know that it can take all kinds of beating. So proteins are excluded in two ways. I'll ask this on the exam. They're excluded in two ways, and that's not in the notes. Proteins are excluded in two ways. There's size exclusion and there's charge exclusion, electrical, right? The glomerulus is negatively charged. Okay, the membrane, basement membrane is negatively charged, so that repels proteins back into blood. What diabetes does is it makes the holes in the capillaries larger. Make sense? Yeah. So diabetes induced damage will damage the size exclusion part of this, but not the charge exclusion. The charge is still fine. Okay? That's still working. That's why your urine is not sludge. Makes sense? Even in a diabetic. Even if you have protein, you will find traces of protein. It's not completely filled with protein. Are we clear? You need to do a lot of thinking for this chapter. Okay? So if you decide that I'm going to study this the night before the exam, good freaking luck. Okay? So there's a lot of thinking that goes on in this chapter. Please pay attention. Please keep up with it. In fact, get ahead of me. Okay? Watch the video and move ahead of me so that when you come here, you're listening to it another time. Okay? And I told you this last week. I told you to watch the kidney videos before you come. Okay? Then fluid moves into the curly coiled up tube, right? And the scientific term for coiled is convolution. Convolution. Convolution means coiled up. Okay? So you have two convoluted tubules. You have the proximal convoluted tubule, and then you have the distal convoluted tubule. They're all labeled. PCT, proximal convoluted tubule. Proximal means you're close to the neck, right? You're close to the head of this thing, right? And then distal convoluted tubule means you're far away. This is a functional distance, because actually the distal convoluted tubule touches the distal is actually closer to the glomerulus than the proximal is. If you took my anatomy, you'll learn that, right? It's actually closer, right? So the name is functional, not structural. So distal meaning the fluid is much farther away because you've gone through the processing of proximal, you've gone through the processing of loop of Henle, and then you hit distal. It's pretty much towards the end of the process, not end of the tubes, end of the process. So distal here refers to functional distal, okay? And then you move into this yellow tube called the collecting duct. And that's pretty much where the filtrate, you absorb as much water as possible, and then finally it becomes urine. Up until that point, so from the start of the nephron till the end of the nephron, this liquid is called a filtrate. A filtrate. It's not called urine. Only when the filtrate leaves the nephron, it's called urine. Are we clear? Because urine means that's it. You can't reabsorb anything else from it. You're done. Right? Urine is what you pee. Correct? Up until the end of this spot, up until the end of this little structure here, up until here, it is called filtrate, not the urine. Please be careful. Because when I say urine, when I say filtrate, it makes a world of a difference. If there's anything beneficial to reabsorb, it's filtrate, particularly water. People often forget water as an important nutrient. Okay? You should not do that with the kidneys. The whole purpose of the kidneys is water balance. Is everyone understanding filtrate versus urine? Okay. So, in between the proximal convoluted tubule and the distal convoluted tubule, you have a tube called the loop of Henle. Loop of Henle. So Henle's great-grandson was my anatomy teacher in high school. So that always makes a special, Mr. Henle. He was my anatomy teacher. So I literally went up to him and I said, did you discover this? Of course, like an idiot. He goes, no. He's like, if I discovered this, you think I'd be teaching here? And teaching idiots like you? And he said, no, it was my great-grandfather. And of course, I didn't believe him, and he had to bring in pictures to show me. And it's a very interesting story. I see everybody cares about this so much. Okay, the interest is overwhelming. All right, so loop of Henle. So you got the loop of Henle. And the loop of Henle's purpose is to reabsorb water. Main job is to really absorb as much water as possible. We're going to be looking into what happens at the loop of Henle in detail. Okay? I think everything before this is anatomy, so you can kind of ignore it. Yeah, most of this is anatomy, but don't ignore it. I would read it, okay? Because a lot of those structures will help you understand how these processing happens, okay? It is really helpful. But I'm going to jump right into the two types of nephrons. So we have two types of nephrons. There are two things that happen in the kidney, y'all. The first thing that happens is you want to reabsorb as much water as possible. Do you agree? It's the whole point of the kidney, right? So we have nephrons dedicated to reabsorbing water. Yes? And those are called, juxtamedullary nephrons. Juxtamedullary nephrons. Medulla is nothing but, the kidney medulla is nothing but a thick salt solution. Okay, the medulla of the kidney, renal medulla, is very concentrated salt solution. What does salt do? It helps absorb a lot of water. Correct? So juxtamedullary nephrons are sitting in heavy salt, right? What will that do? The salt will pull the water out, correct? Guys, you know that's what salt does, right? Salt pulls water out, correct? What? That noise?
No.
Why are you scratching your face for?
Continue.
there's 5 ATPs. I'm never getting that. Okay, ATP waster. So juxtamedullary nephrons. Juxtamedullary nephrons, okay, their main purpose is reabsorption of water. Okay, they help reabsorb a lot of water. Very, very important. And the second nephron type is called the cortical nephron. Guys, bottom line, nephrons in the cortex, nephrons in the medulla. Do you understand that? In the medulla, you're reabsorbing water. In the cortex, where there's a lot of blood vessels, what do you think you're reabsorbing? All the nutrients, right? Yeah, that's why blood is there, exactly. Now do you see how that anatomy helps with this understanding of the physiology here? Cortical nephrons, I repeat, cortical nephrons are meant for reabsorption of nutrients. Cortical nephrons are meant for reabsorption of nutrients. Juxtamedullary nephron is reabsorption of water. Okay. Okay, and that's stated right there. Here comes regulation till your head spins 180 degrees. Okay, get ready. The first mechanism we're going to look at, okay, is this structure called the juxtaglomerular complex. I'm not talking about juxtamedullary nephron. Words seem very similar. Be careful. Juxtaglomerular complex, okay, or JGC, juxtaglomerular complex. You have 3 main cell types here, okay, and this plays a very important role in blood pressure, okay, extremely important role in blood pressure, and we're going to see how, okay. Okay, so the first one is the macula densa. Macula densa. And this is the junction of the ascending loop, okay, and the glomerulus. Now, the ascending loop is basically a distal convoluted tubule. So remember I told you the distal convoluted tubule comes back to the bowl, right? The renal corpuscle or the Bowman's capsule, right? The head of the nephron, right? The distal convoluted tubule comes back to touch it. So that's why I said anatomically it's not far away, right? That junction point is called the macula densa. Extremely important because the macula densa regulates sodium chloride, which automatically regulates blood pressure, correct? Good morning, good night, good afternoon, good evening, good riddance. So everything good. Sodium chloride controls water levels, that controls blood pressure. Okay? Extremely important. DCT stands for distal convoluted tubule. Distal convoluted tubule. Okay? The second cell type is called granular cells. Guys, these are all found together. Okay? This is a structure that is right next to the glomerulus. You remember where the glomerulus is? Yes? It's that bowl, right? Okay. So I'll take you back to this diagram. This is the glomerulus, right? Everyone see that?
Yeah.
Good morning, good evening. Yes. Yeah? Yes. So the juxtaglomerular apparatus is this triangle. Everyone see that? That's the juxtaglomerular complex. Right between the 2 big arterioles. That triangular region. Okay? It has 3 cell types. What are the 3 cell types? Macula densa. Okay? And
granular cells.
Granular cells and mesangial cells. Okay? Sounds like another name I'd give my daughter. Mesangia. Sounds so beautiful. Yeah, it's like I'm so mentally gone. Okay, mesangial cells. Well, before we go to mesangial cells, granular cells are actually modified smooth muscle. The minute I say smooth muscle, what should come into your head? Not the words hell no. But other than that, after that, what should come to your head? Smooth muscle means what? Autonomic nervous system. Thank you. Autonomic nervous system. What's the only autonomic nervous system in the kidney? Sympathetic. So you're only dealing with sympathetic. These are mechanoreceptors. Okay? Mechanoreceptors. So what does that mean? Mechanoreceptors. They sense stretch, correct? You have to know this by now. Mechanoreceptors. Minimus. Don't give me that sarcastic look. Mechanoreceptors means stretching, right? Stretching.
Yes.
This class is gone. Okay, so stretching. Those mechanoreceptors, these release renin. Remember renin? Remember angiotensinogen? Remember that nonsense, right? The RAAS pathway? This is where renin comes from. It's from these granular cells. Now think about how great evolution is, all hail evolution. You're placing these cells in that triangle between two big arterioles, correct?
Mm-hmm.
Think about it. Where else do you want to place it? That's the perfect spot. Because it constantly gives 24/7, 365 the update on what your blood pressure is like.
Mm-hmm.
Look at how amazing evolution has created this, right? That one's a goner, except on job. Okay, so important. So that's granular cells. The third type are your mesangial cells. Mesangial cells, these are connected by gap junctions. Hello, important, right?
Yes.
Gap junctions, which means they're synchronized, correct?
Mm-hmm.
And mesangial cells typically have the ability to pass signals, okay, from the macula densa to the granular cells. So mesangial, the word mes means middle, correct?
Mm-hmm.
Angia means blood vessels, angiogram, right?
Yes.
So what's mesangial? It's found between the two blood vessels, correct? So what do mesangial cells do? They take messages from the macula densa and they take it to the granular cells. So mesangial cells are situated right in between. That's why the word mes. Mes means middle. Okay? So these three regulate blood pressure in our body all the time. And this is a long-term regulation because kidneys take a while to get things done, correct? If you want rapid effects, what would you want? If you want to rapidly regulate blood pressure, you can't rely on the kidneys. Are you clear?
Yes.
And you already know how MAP is rapidly regulated by nerve mechanisms, correct?
Mm-hmm.
Most of the hormonal mechanisms work either mid-level, they're not too quick, but they're not too slow. But if they're working through the kidneys, right, it's going to take a while. For example, ADH is a vasoconstrictor, correct? So the vasoconstriction takes some time because ADH has to travel, bind its receptor, then vasoconstrict. All that takes time, signal transduction, right? But ADH acting through the kidney, what does ADH do in the kidney? It helps reabsorb water. That's going to take a long time. Am I clear?
Yes.
So all kidney mechanisms are very slow, but they have a very big effect. Clear?
Yes.
If you have to build a really small tent, it's going to take you a couple of minutes to build that tent, right?
Mm-hmm.
But if you were to build a 10-story building, that's going to take a while,
Yes.
correct? It's that kind of an idea. Any questions on the juxtaglomerular complex? It's going to become very important in a few minutes. Juxtaglomerular complex. Okay? And if you're interested in the lab, we have a nice model where I can actually show you where this is located. It's very interesting. Okay? Keep that in mind because that's going to come back to haunt us. Okay? So I'm going to summarize urine formation. How do we form urine? What do we exactly do? There are three processes, and this is all color-coded for you. Okay? The first process is filtration. Obviously, you're filtering blood. I told you, right? You get everything into the nephron, and then you pick up whatever you want, right? So filtration is non-specific, and you're going from blood into the nephron lumen. Nothing new. And where does it happen? It occurs in the fenestrated capillaries of the glomerulus. Fenestrated capillaries of the glomerulus. And remember, proteins are excluded by size and by charge. Proteins are excluded by size and charge. Notice how ghetto I wrote that. No proteins allowed. Really? Is this how I speak? You know I'm better. I wrote it like that on purpose because I want you to take notes. So what should you be writing right there? Where it says no proteins allowed, right? You put a frowny face and you say proteins are excluded size and charge wise. Let's go, chop chop, write it. Lazy fools. And then you see it on an exam, you're like, oh my god, I don't understand, I hate this language. I've been screaming this at you for how many times? You need to know this. Proteins are excluded size and charge wise. Okay? And this is meant for you to understand, it's like saying hello, right? After that, you're going to have a conversation. Okay? Charge and size wise is the conversation. Okay? More complicated. What happens with reabsorption? You're being very, very specific. Yes? Makes sense. You want to only pick up things you want. Right? The only exception here is water. Water is always wanted. There is never a physiological situation where water is not wanted. Do you understand? This is why water can kill you. Think about it, right? Remember my story about my frat idiots, right? Where they made the pledges chug water because they couldn't chug alcohol anymore. And then they still died, right? And the reason is, that teaches you a lesson. That means, I don't care how much water you got, you're always going to reabsorb it. Okay? We're desperate. It's like this class and points, right? You want the points. You want the water, always. And there's never a situation where water is chosen against. Are we clear? Because we're land animals. We are land animals. Water is always precious. Even to the point where water can poison you. Right?
Yeah.
No other electrolyte is subjected to that. Only water. So water is always taken up. Tubular reabsorption is very specific with the exception of water and saturable. And you're going from the nephron lumen back into blood. And I want to really emphasize the word back into blood. And I would write that there. Back into blood. Because it is reabsorption. See how it's teaching you? Back into blood, right? Reabsorption. And you're using every possible cell transport mechanism. You are using every possible cell transport mechanism, including molecular and bulk transport. So if you don't remember cell transport, time to review it. Okay? So you're also using transcellular and paracellular modes, and this is happening at the proximal convoluted tubule, the distal convoluted tubule, and the collecting duct. It happens in all of these places. Remember, this is just an outline. Okay? What about the final stage, which is tubular secretion in green? Secretion means you're secreting into the lumen. Secreting always means goes out of the blood, right? Secretion means goes out of the cell or goes out of blood. Here it's going out of blood. You're going from blood into the nephron lumen. And it is also specific and saturable. You're throwing away selective trash. You don't throw away your entire closet, right? You throw away clothes that are torn and you don't use or whatever. Okay? Or it reminds you of your ex. You want to get rid of it. Okay? So, what modes? All modes of cell transport are used here also. Okay? And interestingly, this is transcellular only. This is very, very interesting. Okay? Secretion, you know that's not in the exam. Secretion is only transcellular. It is not paracellular. Guys, you're throwing something away, right? Yes?
Yeah.
If you're going transcellular, things are going to go a lot more slowly. You have a chance of taking it back. Your body is that hesitant to throw things away. Are we clear? Yeah?
Yes.
Remember, so question is, well, why would you ever want urea back? Remember, urea is not secreted. Urea is filtered and never reabsorbed back. Are we clear?
Mhm.
Here, you're actively throwing something into the trash. Am I making sense? There's a difference. Urea gets filtered at the filtration stage and never reabsorbed back. Whereas tubular secretion is what? You're putting something actively into the trash right before the trash man comes. Am I making sense? Right before the truck shows up. Okay? So, you want to make doubly sure you are throwing it away. That you don't want it, right? Because once you put it in, you can't take it back. Make sense?
Yes.
So, that's why it's transcellular only. It's not paracellular. Transcellular is a lot slower, and it might be useful for the cell, right? So you have to go through the cell. Many times, what's secreted is usually charged. Many times, these are ions, like potassium. Okay? Is there a time of day when you need potassium? Sure. Potassium becomes valuable, right?
Yes.
So you always go through the transcellular pathway for that, okay? This also happens at the PCT, DCT, and CD. I want everyone to look through these three processes and ask me any questions.
So, filtration is happening like all the way up into the blood in there as well.
No, filtration only happens at the glomerulus, remember? See, did I say that? No, I didn't say that. No, I did say that. Another 5 ATPs gone.
I have a question, like, when somebody, they have caffeine intake, and it has enough time for the kidney to filtrate, like, the digestive water.
So the question is caffeine, okay? I have to look into this because I don't know the answer. I don't know if caffeine is filtered into the kidney. I would think it would be because it's small, right? But I know caffeine gets absorbed through your digestive tract, right? Nobody injects an IV of caffeine, right? So remember, a lot of this is bloodborne, correct? Yes?
Mhm.
Nobody injects caffeine into nobody does, right? I mean, I'm going to evolve to that point. So, but you don't inject, so it won't be in your blood. It's usually digested, right? So, but let's say that caffeine gets absorbed into blood from your digestive system, right? The amount there is going to be relatively small. Now, does that caffeine amount filter through? I don't know. But my answer is yes, it does. Okay, it most likely does. Okay, but I have to check if it does or not. Okay?
Doesn't everything the digestive system absorb is at least carried through blood? To
Yes,
the
but the amount of caffeine that's absorbed is not going to be 100%.
Not just caffeine. Oh, okay.
It can be,
Yeah.
right?
Mhm.
So a very small amount of it gets absorbed
Mm.
and transported through blood, but you don't know what kind of proteins it binds to, and, right? We don't know the kinetics.
Okay.
So it's a very important question, right? So that's a really important point because this is the same fate for all kinds of drugs, right? When you take any kind of drug, it's silly to think that 100% of that drug is going to be in your blood. It won't, especially if you take it orally, right? If you're injecting it, it's different,
Mhm.
right? But if you're taking it orally, very little of it makes it in your blood, usually.
Actually, I have a question because when we take the caffeine, it's gonna like attack to the medulla of our brain, and then it would inhibit the ADH. And when the water getting filtrated, that is the water already in our blood, or that is the water.
So Yeah, I'm gonna stop you there because there's a flaw in what you're saying. So, what she's saying is caffeine inhibits ADH, it doesn't. Alcohol, my other favorite, inhibits ADH, okay? What caffeine does is it causes your bladder smooth muscle to constrict. And that's what makes you pee. That's why after coffee, your pee volume is probably really small, right? Because your bladder is not really full. But caffeine will signal, right? It will inhibit phosphodiesterase and it will increase cyclic AMP levels, which causes smooth muscle contraction in the bladder. And it causes you to pee whatever little you have out. Okay, it's very important because it's important to understand that think about it, and I'll tell you why the reasoning. If caffeine were to inhibit ADH, caffeine should make you sleepy. Correct? Because what does ADH do? It's a vasoconstrictor, right? Right? So when you relax your blood vessels, what's going to happen? Brain's going to receive less blood because gravity is going to pull the blood down, correct? And this is why alcohol makes you sleepy. Yes? Finally. So that's why caffeine cannot be doing that. Okay? I don't know that for a fact, but hypothetically it makes sense that caffeine should not be doing that. If anything, caffeine should be vasoconstricting, pushing more blood into the brain. And that's why brain throws a tantrum, right? Brain spoiled like hell, right? Like Francesca over here. So, your poor brothers. So, brain's really spoiled with all that blood. So when you don't drink the coffee, what happens?
Angry.
Brain throws a bitchy tantrum, correct? Where is my coffee? And if you see me at 7:30, you'll know what that looks like. Okay? So, I'm all nice and awake because I keep drinking my little cafe latte protein drink from Costco. Where's Angry Birds? Yay, I should have brought one for him. Okay, so are these three processes making sense? Yes? I don't know what's making sense, what's not. But moving on, now let's go into the details. Yeah. Just like in respiration, where you had a respiratory membrane. Yes?
Yes.
Ah, what the hell, 5-minute break. So that one, what is happening there is what we call cardiac toxicity, where the heart can't take it. So in other words, so what's happening is the vasoconstriction is so strong that the heart receives so much blood, and the vein-bridge reflex stops. The sympathetic system slow turns on, the heart's going crazy, a lot of blood, the whole thing just comes to a crashing halt. And the parasympathetic will block the heart. Right? Because remember, sympathetic goes way too high, the parasympathetic will block it, and that's why you faint. Right? But that fainting couldn't go into a coma if you don't get enough blood to the back of the brain. So that's what happened. They initially fainted, and then you didn't get enough blood back to the brain, and they went into a coma. And then death. Those are the stages. Okay, so fainting is me looking at your test score. And then when I look at it carefully, see what things people got wrong, then I go into a coma. And then when I have to do your final grade, I want to die. So that's the stages. No, this is exactly the kind of way you should think about physiology. Taking all these gossip and then making sense. I mean, you're making logical sense of it. Because one day you're going to get patients like that. Yeah. But that's what must have happened. Excessive caffeine will speed up your heart so much that the brain will put the parasympathetic brake on it. And normal people will usually faint and recover. Right? But if you fainted so much that, or the parasympathetic brake was so strong that the brain doesn't get enough blood in the right amount of time, the brain will go into coma. And coma to death is not that far of a distance. Yeah. Why did Angry Bird fly the cuckoo's nest? Can't take it no more.
He was getting tired.
That's right. He'll be running after chickens to put in the blender.
Told me about a sandwich where instead of bread for his steak sandwich, he used ribs as the bread. Like rib steak.
How can you say these kinds of things to a vegetarian?
I did that.
He's harassing nutritionally. So is this making sense so far? It's a lot of detail.
I mean, it makes more sense as like I reviewed it last week. Or previewed it last week.
And Steven didn't go into all this.
Huh?
Steven didn't go into all this.
Honestly, I can't remember anymore. It's been a while.
What happened when you go get out of college like man?
Got a good grade though.
I used to teach there, though.
Oh, really?
Yeah.
It's a nice campus. It's very nice. The view is so great.
It's a good school. It's not bad.
Yeah. So top of a hill, so like you can see the entirety of Oakland and like South Bay.
Okay, I can't stand no more. I have to sit. Grandma's tired.
It's a month about here last semester.
So people who came in late, just letting you know that next week, lab session is optional. You don't have to come. Okay? Translation, show up and die. Okay? No, just don't waste your time. Okay? Use that time to study and just show up to lecture. Okay? That gives you a couple more hours. Right? It's up to you. If you want to come, I'm going to be in the lab. So I'm not going anywhere, and I have a 7:30 class, so it doesn't make any difference to me. I have to be there anyway. So I'll be there Tuesday and Thursday. So feel free to come. So if you want to come sit and study with me and you want to ask me questions, that's fine. But otherwise, yeah, you don't have to come. Just make sure you come to lecture, because we'll be there in fluid balance. Okay, so let's go through these three processes in detail. So most likely I'll get through glomerular filtration, but that's all I'd probably do. And I may not even finish this. Okay, so what is happening at the glomerulus? Guys, you have to remember the structure of the nephron for this. Please, this is really, really important, or this stuff won't make any sense. We're right here, okay? We are right here. Everybody got that?
Yes.
Okay, for the next 45 minutes. Okay, so you have a filtration membrane, just like you have a respiratory membrane. Remember, you have the respiratory membrane with all the alveoli, right? So similarly, you have a filtration membrane, and the filtration membrane has the following. It obviously consists of the endothelium, and if you recall, endothelium is the cell layer of the capillaries, right? Remember, capillaries have only one component, right? You have cells, flat cells, because capillaries are supposed to do what? Simple diffusion, right? So you have simple squamous epithelium, which aids in diffusion, right? So the filtration membrane consists of one layer of capillary endothelium, okay? The basement membrane, remember that negatively charged membrane that I kept yapping about? That's called the basement membrane. Basement membrane, when cancer spread, they break the basement membrane. Every organ has a basement membrane, are we clear? It's found throughout the body. Every organ has it, okay? So, basement membrane, and lastly, you have the foot processes. They literally look like feet. Foot processes of podocytes. Who's a podiatrist? Who's a podiatrist? Foot doctor, right? A doctor who treats problems with feet. And in case someone's wondering, who the hell has problems with feet? Diabetics do, right? Neuropathy, right? So, a lot of times people think, same with dermatologists, right? They call them pickle poppers, right? People forget skin cancer, right? So remember, people don't just do MDs for no reason, right? So podiatry is important too. So podiatry is medicine of feet, right? So here, podocytes are cells that look like feet. They're incredible. They truly look like feet. And you know what they do? So here's my foot. Now I'm gonna die. Okay? So if I push my foot, right? I want you to imagine quickly before I sprain every muscle in my body. There's the glomerular capillary at my feet, right? Glomerular capillary at my feet, everybody imagining?
Yes.
Yeah? So if I push my foot and you push your foot, we're squeezing those capillaries, yes?
Yes.
Yeah? So if we play footsies, right, and the capillaries are in the middle, what is that going to do? It's going to squeeze the capillaries. It maximizes your filtration rate. Yes?
Yes.
So now do you see why they're shaped like feet? To have really high surface area. Make sense? So you squeeze the capillaries. So these cells are constantly doing this, right? Which means they need calcium. If cells need to move, they need calcium, correct?
Yes.
Okay, muy importante. So all three of these comprise the filtration membrane. So I can easily ask you a question. Which of the following is not a component of the membrane? Or which of the following is dependent on calcium for its function? That would be the podocytes. Are we clear?
Yes.
Okay. Then we got the fenestrated endothelium. I've already talked about this, right? You have holes. You have a size barrier, right? So anything smaller than a protein gets through, and the basement membrane is negatively charged, right? And it's an electrical barrier to proteins, okay? And this membrane enables you to retain large macromolecules in blood. And here I primarily mean proteins, but there's carbohydrates and there's some large molecules there, which help pull back water. Pulling back the water, is that important or not? Yeah, right? So you've seen this equation before, correct? NFP, net filtration pressure, is hydrostatic pressure minus osmotic pull minus the capsular pressure. Don't worry about the capsular pressure, but this number I need you to memorize. And you've already seen this before. 10 millimeters of mercury, amount of fluid goes into the nephron. Are we clear?
Yes.
Okay? This is the purpose of next week's lab. That is the purpose of next week's lab, which means a minimum of 3 questions on your next lab exam. Okay? So the radius of the afferent arteriole, at any given time is bigger than the radius of the efferent arteriole. Now I want you to think. So in other words, if I have to draw this, right? So here is the entrance to the nephron. Everybody see that? In blue, yeah? And here comes the afferent, and here is the efferent, right? Everybody see that? Yeah? Question is, why is this so much bigger and why is this so much smaller? Because a lot more blood comes in here, correct?
Yes.
And sludge leaves here, correct? You filtered everything, correct?
Yes.
Make sense? So you need that to happen. I'll just draw this permanently. It's kind of important. So do this in red. So you have a large afferent and a small afferent. Yes? So that's what it looks like. It's very simple. Afferent brings in all the water, right? And the glomerular capillaries take out all the water. So what's left?
More water.
Sludge,
Yes.
right? And if sludge has to go through a vessel, and the vessel is huge, what's going to happen?
Spending time.
Sludge is going to sit there. It's not going to move. So you need that efferent arteriole to be really, really small. So the sludge can be squished through. Guys, I want you to imagine this. Let's say you have a huge amount of toothpaste filled with water. It's a lot of water there, right? So the toothpaste has to go through a filtering system, and then what comes out is this gelatinous paste, right? If the gelatinous paste sits in a big tube, it's not coming out, right? You want to squeeze the tube for the paste to come out, right? What are you doing? You're making the radius a lot smaller, right? Am I making sense?
Yes.
Yes, Joe.
But would it decrease radius also increase resistance?
We're going to get to that. So the question is, wouldn't decreased radius mean higher resistance? It's going to be higher resistance no matter what, because viscosity is out the roof. Am I echoing? Or is it me? Okay, my ears are busted. What's going through the efferent arteriole? That's a very, very important question. What's going through the efferent arteriole? Sludge. Is sludge viscous?
No.
Yeah.
Well,
Sludge is
thick. Sorry.
extremely thick, right? Sludge is like honey, right? Very, very viscous. Sludge is the maximum viscosity you can have, correct? You've lost all the watery portion, correct? So yes, extremely high resistance. At any given time, the efferent, make sense? Although the efferent arteriole is a lot longer because of all the peritubular capillaries. So that also adds to the resistance, technically, but I'm not going to hold you to that. Is this making sense?
Yes.
If you do not write all this down, and if you do not follow up tonight by looking at this lecture and learning this actively, kidneys are gone. They're a hopeless chapter for you. This is a very, very important chapter for any medical profession. Okay? So please, please invest time in this. I have never said this about any of the other chapters. I didn't even say this about cardiovascular. I'm saying this about kidney. Extremely important and complicated. It'll get real complicated real fast. So pay attention. And as soon as you move out that door, everything you learned here is gone. Okay? So you have to reinforce this at home. Are we clear? You must study this tonight. Must. Okay. So another important idea, if the afferent, and you're going to be investigating this completely on PhysioX next week, if the afferent arteriole is big and the efferent arteriole is small, okay, what that does is it squeezes the glomerulus a lot better. Make sense? So the idea is there's a resistance gradient, right? You have less resistance in the afferent arteriole and you have more resistance in the efferent arteriole. So in the glomerular capillaries, this increase in resistance will help with the filtration process. Very simple example. Everybody's seen this, right? When you want a lot out of your tea bag, let's say it's a very expensive tea bag, right? I mean, I got this rose flavored tea from Canada. This thing was $25 for 10 tea bags, and believe me, it was worth it. So it's black tea infused with rose petals, right? It's amazing taste, right? I squeezed the damn thing until the bag tore. Okay, but that's just me because I'm brown. But the point is, everybody squeezed a tea bag, correct? You wrap it around and you squeeze it. That's what you're doing here. That increase in resistance squeezes that tea bag. Make sense?
Yes.
To get as much fluid out as possible. Everybody clear about this? So everything has a purpose. You just have to look for it. Everything has a purpose. Okay? So glomerular filtration is proportional to the following. The net filtration pressure, right? The more filtration pressure you have, the more you're going to filter. This is why people with hypertension, kidneys are the first things to go. Because even before the heart, Because if your blood pressure is always going to be high, guess what? Your filtration is always going to be high. Correct? And you have little holes, right? If you keep on putting pressure on those capillaries, the holes are going to become big and they're going to break. Okay? Very important. And number 2, surface area. Okay? And this is something you really have to imagine with me. Remember the juxtaglomerular complex, that little triangle? Everybody remembers this triangle, right? So it's an upside down triangle like this. Everybody look at my fingers. Upside down triangle, right? So this finger is afferent arteriole, this finger is afferent arteriole, right? So when the mesangial cells constrict, what do? They bring these two closer together. When that happens, you put pressure on the glomerulus. Are you visualizing this?
Yes.
These are the mesangial cells. When they contract, what happens? Remember, they have gap junctions, right? So when they contract, they're pulling these blood vessels closer. When they pull these blood vessels closer, it's putting pressure here. Okay? To filter much better. Exam question. If mesangial cells fail to function, which of the following is going to be massively affected? And the answer is filtration. You're not going to be filtering as well as you should. Okay? And they're all regulated. Okay? And lastly, the fenestrations, the actual holes. Okay? So what are we talking about? What affects glomerular filtration? Your blood pressure is the number 1 factor. The second factor is surface area, right? If I'm going to squeeze that triangular area, it's going to put more pressure on the glomerulus. That's why it's strategically located there, right? And third, the holes. Think about it, if those holes are all filled, right? That's what happens in diabetes. The holes become calcified because you have so much sugar there, right? The holes fill up, right? And when the holes fill up, right, what's going to happen? The capillaries are going to expand, right? Because water's not getting through. They can burst. Make sense? Kidney damage. Are we clear?
Yes.
Right? I mean, that's of course a very severe portion. So how do you regulate GFR? What does GFR mean, first of all? Glomerular filtration rate. Glomerular filtration rate. Okay? I think I've put the kidney flow chart on your modules. If it's not there, let me know. I don't even know if I have a kidney flow chart from Amna, but I know she made one or two. This were useful. Anyway, how is GFR regulated? I guarantee you, minimum of 10 questions are going to come from here. Minimum, bare minimum, 10 questions. There are multiple ways of regulating glomerular filtration rate. So what are we talking about? Remember that bowl? Yes? Filtration. How fast is filtration happening? How do you control that? During the day, you can increase it or decrease it, right? Based on stimuli. Question is, what is the stimuli? Second of all, what are the stimuli? Second of all, how is it regulated? So, the most important mechanism is an auto-regulation. Auto means what? Self. Auto means self, right? Okay, auto means self. And this is something you've already seen before, many times over. Okay? Arterioles contain what kind of smooth muscle? Multi-unit. And when multi-unit stretches, what happens? It snaps back, right? So when too much blood gets into the afferent arteriole, what will it do? It'll expand and it snaps back. Okay, why? Because when that much blood goes into the glomerulus will explode and damage the nephron. Are we clear? So it prevents that damage by clamping. Okay, and stopping blood from going, all the volume going into the nephron. It's very nephroprotective. And if you have too much blood, AA is afferent arteriole will constrict, correct? Preventing high volume from bursting the nephron. Is that clear? The second feedback is a long ways around. It's called glomerular feedback. Break the words down. Tubules, glomerulus. So the tubules affecting the glomerulus, right? So here, what happens is the following. Guys, one of the reasons why this chapter is so difficult is people study this in bits and pieces. You don't connect it all. Every 2 seconds you have to ask, what's the big picture? For example, when I'm at the tubular-glomerular feedback, I would stop and say, okay, what am I studying here? I'm looking at how is the filtration process regulated, correct? So in the first pathway, where it's myogenic, right, what's going on? Glomerular filtration rate goes down when too much blood gets to that area, correct? Second is tubular-glomerular feedback. Now, before I go into this mechanism, think common sense. If your tubules are really, really full, do you want to decrease filtration or do you want to increase filtration? What do you want to do?
Increase.
The tubules are already full, like me,
Tubules.
right? You want the tubules to burst?
No.
No, you want to stop filtering, right? First clear this fluid, and then you can filter. Am I making sense?
Yes.
So these are both GFR decreasing mechanisms. See, that's why it's important to go back and zoom out and ask yourself, what the heck am I studying here? It's nice and so interesting. Okay.
Arterioles go to the tubules. Those
Glomerulus,
are
yeah.
the ones that have the blood. The afferent one especially is the one that has the filtrate or blood.
So the afferent arteriole brings blood
Okay.
with all the water and all the goodies
Mhm.
into the glomerulus. The glomerulus is the key back, that's where you're filtering.
Yes.
And the afferent takes all the sludge back.
And that sludge is as
All cells
it
and
becomes-
proteins,
Yes.
anything bigger than the whole.
Whereas the smaller things are become part
Have
of the
gone,
filtrate.
yeah, have gotten into the
Okay.
filtrate, exactly, into the nephron. That's an extremely important point.
Mhm.
So if GFR is really high, y'all, what happens? The tubules are going to be beyond full, correct? And so what happens is, if there's going to be high filtration rate, fluid's going to move fast or slow through the nephron. Really, really fast, correct? And if you're moving that fast, can you absorb things quickly?
No.
No. Remember, slow movement means maximum absorption. Slow movement means maximum absorption. Not too slow, but relatively slow movement. So what you need to do is, so what happens actually, is you have very little time for NaCl to be absorbed, right? Because NaCl goes transcellularly, right? And sodium will never go paracellularly. Very important. Sodium is too precious. Am I making sense? Plus, if sodium goes through paracellular, that will electrocute the kidney. Am I making sense?
Yes.
Hello? Because it's a very charged dense ion and it's moving really fast, right? You will cause huge electricity there. So sodium can never go through paracellularly. Please note that down. Sodium will only go through transcellularly, always. And plus, sodium is coupled, remember, to secondary active transport. Remember that? Everything piggybacks on sodium. Do you have sodium that you can afford to go through paracellularly with nothing on its back? That's stupid. Every sodium going through should be piggybacking something. Do you understand?
Mhm.
Okay? So if GFR is high, your fluid flow is going to be really fast, correct? Which means your sodium chloride is not going to be reabsorbed. Do you agree? Now, the macula densa senses a lot of sodium chloride. Macula densa is all chemoreceptors. Macula densa is chemoreceptors, and the sodium chloride is sensed by the macula densa, and sodium chloride will immediately, okay, release ATP. Here, ATP is not used for energy. ATP is used as a neurotransmitter. Very, very important. you release adenosine. Adenosine is ATP, right? ATP is released. Now do you see why the kidney is red as heck? It needs a lot of mitochondria to make all these ATPs. Okay? ATP is released. ATP will immediately cause vasoconstriction. And will vasoconstriction bring down GFR or raise GFR? Vasoconstriction, will it increase GFR or decrease GFR? Decrease GFR, is that what we want? That's tubulo-glomerular feedback. Because ATP is released by the distal convoluted tubule. The tubulo-glomerular feedback is the distal convoluted tubule, not proximal, distal. Tubulo-glomerular feedback is the distal convoluted tubule, okay? And you vasoconstrict the afferent arteriole, okay? And GFR goes down. What type of feedback is this? Negative. Now what if your GFR is low? Right? So right now, because I didn't eat, my sodium levels are probably low, right? And if sodium levels are low, my water levels are probably also low, right? So my blood pressure overall is low. If my blood pressure is low, what will my glomerular filtration rate be? If my blood pressure is low, what is my glomerular filtration rate?
Low.
Low. Am I making sense? This is why intermittent fasting is very, very protective of the kidneys, because it makes kidneys get the rest they need. Am I making sense? Correct any other metabolic issues you have in the kidney. Very, very beneficial. Again, a lot of traditions do fasting, and fasting is very beneficial for the kidneys. It's beneficial for the whole body, but usually when the kidneys benefit, the whole body benefits. Okay? So GFR is low, the fluid flow is low, right? Which means you have a ton of time to reabsorb sodium chloride. I'm already sodium chloride deficient, right? So I'm going to want to reabsorb a lot of sodium chloride. Macula densa, in this case, will release renin. The same macula densa released ATP when the pressure is too high, correct?
Mm-hmm.
When the pressure is too low, it will release renin, and you know what renin does, correct? Right? Renin angiotensin, remember? Renin will increase angiotensin. Angiotensin will increase blood pressure, right? And here's the really interesting part. Macula densa will also release prostaglandin E2, PGE2 is one of the worst parts of intermittent fasting, is all the burping, particularly this time. I'm like a regular Thomas the Tank Engine. Okay, PGE2, right? And nitric oxide, our favorite nitric oxide. What do you think that does? Don't think gives you an erection. Yes, it does, but not in the kidneys. If you have an erection in the kidneys, we've got trouble. Okay, so PGE2 nitric oxide will vasodilate, correct?
Mm-hmm.
It will vasodilate the afferent arteriole. And here's the interesting part. You know what angiotensin does normally, right? It vasoconstricts, correct? Okay? As far as I know, and I'll have to verify this, but as far as I know, angiotensin does not affect the afferent arteriole because there are no receptors. It only affects the efferent arteriole, and efferent arteriole is constricted. Remember, no receptors, no effect. Correct? Because think about it. If afferent arteriole had angiotensin receptors, you'd be squeezing the afferent and you'd be squeezing the efferent. That's going to make your glomerulus explode, right? It's not going to help. Do you understand the physiological disaster there? You don't want that. Okay? So you dilate. When you dilate the afferent arteriole, you increase blood flow. Makes sense, right? Your GFR is low. So what should you do? Increase GFR. How do you increase GFR? Make more blood flow there by decreasing the resistance, correct? How do you increase the resistance? By increasing the diameter. And think about it. If I'm making the afferent bigger, right? Think about it. If I made efferent also bigger, what would happen? Everything will collapse, right?
Yes.
There's already not enough glomerular filtration, right?
Mm-hmm.
go back, zoom out. What was the initial problem? GFR is too low, which means you're not filtering. If you want to filter, what do you want to do? You want to bring more blood in and squeeze the glomerulus more. If you want to squeeze glomerulus more, you want to squeeze the efferent arteriole more. Make sense?
Yes.
So the glomerulus experiences more force and will filter better. So know this, and you're going to do this in PhysioX next week. When you increase the diameter of the efferent arteriole, more blood comes in. That should be common sense. More blood comes in. When you squeeze the efferent arteriole, when you vasoconstrict the efferent arteriole, you're putting a lot of force on the capillaries, right? And that will help filter more. So to decrease GFR, what should you do? You should dilate the efferent. Am I making sense? Release that pressure. Okay? And if afferent and efferent both were the same radius, you're doomed. There will be zero infiltration. Are we clear?
Yes.
We do not want that. Make sense or not? So what was the original problem? GFR was too low. What is your response? You dilated the afferent, bring more blood. You constricted the afferent, which increases the squeezing of the capillaries. So you bring more blood and you squeeze it more. Will you get more filtration?
Yes.
Yes, and that's exactly what we need. Good morning, good evening, good night. Does that make sense?
Yes.
And that's what's shown here. Okay? So you need to know very little of this. You need to know very little of this. So note down exactly what you need to know. Those are real cells. It's from a fluorescence microscope. Okay? I took it out of a paper. I can't remember who. But EA is efferent arteriole. AA is afferent arteriole. Notice the afferent arteriole, and this is the glomerulus right here. And this is the macula densa. Okay? So afferent arteriole is a lot thicker. Look at how thin the afferent arteriole is. Everybody see this? Hello?
Yeah.
See this? See how thick this is, right? And some of it's architecture to begin with. Okay? And so what happens is, this is the tubular salt sensing, right? So if there's too much salt, what's happening? There's too much water. And if there's too little salt, you release something else. If there's too much salt, you release adenosine. If there's too little salt, you release renin, correct? And this shows how this works. So sodium and chlorine and chloride and potassium are co-transported. All of them are co-transported here. Yeah? So sodium, chloride, and potassium symported. Isn't this amazing? You've never seen this before. It's a quadruple symporter. Do you see this? And so they all go in, right? And when this goes in, and here's an antiport mechanism, the hydrogen is pumped out, sodium is going in. But bottom line is what happens is this going in will increase PGE2. Okay? So this one is sensing the amount of salt, right? And what happens is when salt goes in, when sodium goes in, hydrogen comes out, right? You're pumping hydrogen out, okay? So it's an antiport mechanism, and these are all channels. Okay, they're all channels. They're sensing these. So when the sodium and the potassium are going in, they're balanced by the chloride, so you don't have action potentials. Are we clear? The voltage is cancelled out. Sodium is +1, potassium is +1, and chloride is -1, right? You take 2 chlorides in. So there's no electrical change. So what happens is you activate these enzymes. COX-2 is cyclooxygenase 2. And that's what aspirin inhibits, cyclooxygenase, okay? So this will cause PG, prostaglandin E2, and that will cause, remember the arterial smooth muscle here? Okay? Cyclic AMP and calcium to go up and all that, right? And you're going to have a baro reflex. Baro means pressure, blood pressure, right? So, and you basically, what you do here, so here's angiotensin 2, right? And here's AMP, these are receptors for these, okay? and you will end up, so here's again the renin granular cells, right? So the bottom line of this picture is showing you how the tubule cells can control the contraction of these muscles. Okay? So remember what I told you, efferent arteriole, right, will vasoconstrict, correct? Now, if sodium is high, right, what happens? You want the afferent arteriole to constrict, right, to decrease GFR. So if sodium is high, it's going to get in, right?
Yes.
Right? And this is going to affect the afferent arteriole and cause it to contract. When afferent arteriole contracts, blood flow is less, your filtration is less. Isn't that what you want to do when sodium is high? Correct? Add to this mess adenosine. Okay, adenosine is not mentioned here. Add to this adenosine. Okay, adenosine is the main factor here. The reason it's not mentioned is because this paper focused on ANP and angiotensin II. Okay, so that's why they're not really talking about it. And what happens is once you constrict this, you have GFR going down. But if sodium is low, right, you inhibit this mechanism. Are you seeing that? Okay? And what ends up happening is, in this case, a hydrogen comes out, okay? And a sodium, and whatever sodium is there, when sodium levels are low, that will activate. So this inhibits NOS, right? If sodium is high, this is going to be low. If sodium is low, this is going to be high. Nitric oxide comes out and relaxes the afferent arteriole. Okay, so this diagram will not be on your exam. I explain it because I want you to get a taste for scientific research. This is how people do research, right? So they look at what actually happens at the cellular level. It's really important to do that. So what will you be tested on is what I wrote. Make sense? This is how, I want you to see how complicated this is. It's not that simple, even though I wrote it in such a simple way. Okay? So I will not ask any of this. Okay, so don't worry about this. What you need to know is here. This you need to know. This one, the GFR low and the GFR high. I need you to know both of those responses. Everybody got that? Okay, so that will stop. We'll continue with, I told you I will not finish GFR regulation. So we'll continue with more GFR regulation on Thursday. Okay, please go through these notes completely before you come on Thursday. The whole chapter. It will help.
Yes.
Okay? Take your time and do that, alright?
Yes, sir.
I will see you on Thursday. And people who are late, let me give you the announcement that I will be dropping your lowest lab exam and your lowest lecture exam. I'll update you on the extra credit exam. Okay? Have a good one.